Welcome, everybody. This is a respiratory cell and molecular biology journal club that we've organized to highlight a current publication in the Red Journal. And we kindly have the group who performed the study and the publication here presenting from Germany. And so I'd just like to present Anna-Lena Ament. So she is the primary author for this study, and she is started her training at the University of Gießen under the mentorship of Suzanne Harold, and currently is continuing her work under her mentor, who is also here with us today, Dr. Ivonne Vazquez Armendariz. And so without further ado, Anna-Lena, if you want to introduce your topic and your title and get started.
Thank you. So first of all, before I start, I would like to say thank you to you, Erin, and to all the organizers for reaching out to us and giving us the opportunity to present our data before it's printed. So we're super excited that it's going to be printed next month. So whoever is interested after the presentation can have another look on the paper. And with that, I would like to start. So our paper is about endothelialized bronchioalveolar lung organoids to model endothelial cell response to injury. And I'm very sure that you all are very familiar with the term organoids or like lung organoids in general, but I would like to give you a brief introduction onto that because there are quite a lot of different models, especially there are a lot of different models about lung organoids, so depending on the stem cell source. So organoids are stem cell-derived, self-organized, three-dimensional tissue cultures, and they can replicate either like the whole complexity of an organ or just selected parts, like really just like specific regions, and they allow a detailed view on organ development and disease. And as I mentioned, there are several stem cell sources that you can use to generate organoids. So you can either go for fetal stem cells, for adult stem cells, or iPSCs. And then, of course, you can use either marine stem cells, human, or any other species. And I will talk a bit about marine adult stem cell-derived organoids because that's what the project is about. And the cool thing here is that there are so many different kinds of lung organoids, which you can derive from mouse cells. So you can recapitulate either the proximal part by using basal cells. So if you put those ones into madrigal, you end up having tracheospheres, or if you go a bit more down and use club cells, you can generate bronchiospheres. Those ones are actually quite funny and cool because they have a lot of goblet cells and also a lot of ciliated cells. So if you're lucky and you look through the microscope, sometimes you can see the cilia beating and then the mucus is like moving around in the organoid, and that is kind of cool actually. But you can also model the more distal part of the lung. So you can generate alveolospheres by putting type 2 cells into culture, and then you end up having alveolospheres with both type 2 cells and type 1 cells, which is pretty cool because in the human system, it's always a pain to have type 1 cells in there because it's quite difficult to differentiate. But for the mouse ones, it works quite well. But there's actually a very cool system which we've been working with, which are derived from bronchioalveolar stem cells, which are located in the bronchoalveolar duct. And if you put those ones into madrigal together with feeder cells, you can end up having different kind of organoids. So you can either have like bronchial organoids, alveolar organoids, or the most interesting one, the bronchioalveolar organoids, which recapitulate both parts of the lung. So you have the alveolar compartment, but you also have some bronchioles. So they are super great when you want to study like the organ development or diseases if you don't need like just a specific region of the lung. So yeah, we've been working with those ones. So with the bronchioalveolar organoids, but I will talk about them in a minute. So before I dive into our data, I would like to talk a bit about the endothelial compartment of the lung, because it's quite special and it plays a crucial role in lung development and disease. So the lung is highly vascularized, and as I said, its endothelial compartment is really special because it has to facilitate the gas exchange. And deficient development of the lung vasculature contributes to chronic diseases after birth, and even after birth, the lung endothelium plays a crucial role in homeostasis and host defense and repair. So, for example, they play a crucial role in the leukocyte trafficking, as you all know, and yeah, they're really important. So recently there have been a couple of studies that identified two different populations of pulmonary capillary endothelial cells, which are called Aridgides, short ACAP, or general cells, short GCAPs. And the ACAPs are marked by a high expression of CAR4, and they are very special because they're specialized for the gas exchange. So they align with the type 1 cells of the alveoli, and they are responsible for the leukocyte trafficking. And the GCAPs, they are marked by expression of apelin receptor, and they maintain the alveolar endothelium, and they also may act as progenitors. So during the last years, there were several studies on generating different kinds of lung organoids, human and mouse, but most of them didn't have like an endothelial compartment, or there were really just a few where they actually had like endothelial cells in there. And the issue is that in all of these models, it was never shown that the endothelium actually interacts with the epithelium. They were only used as like feeder cells, and most of the times they either died over time, or they just like sat in there, like in the matrix. They were super round and yeah, basically not doing anything. So they didn't interact with the other cells. And as the lung endothelium plays a crucial role in development and disease, we thought, okay, we have this cool organoid system. Why not just give it a try and add endothelial cells on top? Can't be that difficult, can it? Well, it turned out it's not that easy, but yeah, I will tell you more about it now. So the organoid system we used was established by my supervisor, Ivonne. I'm doing her PhD thesis. So what she did was she used the lung homogenate of adult mice, and then she went for cell sorting, and then she sorted two populations. So resident mesenchymal cells, which were like CD31, CD45 negative, EpCAM negative, and SCAB1 positive. And then we sorted a population which is enriched with Vazquez. So we sorted for EpCAM high, CD24 low, and SCAB1 positive cells. And then we put both populations into Moderator cultures, and we keep them in insert. So we do have an air-liquid interface. And after 21 days, you end up having like very mature organoids. And this is what you can appreciate here. So this is a video of day 15 to day 20, and you can really see how the organoid forms and how it branches. And here you can really see the formation of the tube. So you really have also the bronchioles. To give you a bit more detailed view, so here we have an overview of an organoid. And region 1 is the distal region, so the alveolar region. And here you have like, you have type 1 cells, which are kind of thin and elongated. And then you have the type 2 cells marked by the expression of surfactant protein C. And then here in region 2, this is the proximal region. So the bronchiolar region, and here you have a lot of club cells and ciliated cells, and you can also see the cilia bleeding sometimes. So this is really great to see. This is really cool. And then Yvonne characterized the organoids a bit further. So what she did was she went for single-cell RNA-seq, and as I mentioned, we also have like a mesenchymal compartment in there, and those resident mesenchymal stem cells differentiate into myofibroblasts and lipofibroblasts, and they play different roles. So the lipofibroblasts, they promote the epithelial growth and dedifferentiation into type 2 cells, and the myofibroblasts, they deposit the matrix, which is crucial for the alveolarization. So they are necessary to provide the scaffold for the alveolarization. So yeah, this is then the model we ended up with. So we have a lot of alveolar stem cells, so alveolar cells. So we have type 1 and type 2 cells, but we also have the airway cells, basal cells, and also some cycling cells in there. And that's where my project started. So I started this project as my master thesis, and after 1 year of work, we were lucky enough to have some nice results. And then I ended up working for a couple more years on this project to really make it work. So I started with isolating endothelial cells from very young mice. And what most people do is when they isolate endothelial cells is that they go for magnetic bead sorting. So they either use like CD31 or CD102 or a combination of both. So I tried that. But to be honest, it didn't really work for me. So I struggled quite a lot. And I always had a lot of contamination from fibroblasts. So at some point, they always overgrew my endothelial cells. So I was quite frustrated, I have to say. And then I went into literature and I found a paper where they actually flow sorted endothelial cells. And I was quite surprised by that, because I've been always told, okay, there's no way you can sort endothelial cells because they will die. So I was really surprised to see that. And then as we anyway had a sorter in Gießen in our facility, I thought, okay, why not give it a try? And then, yeah, I tried the protocol. So I isolated the lungs from PA to P14 pups. And then there's like one major thing. So you can't sort endothelial cells directly from the digest. So there's no way you take out the lungs, you digest them, and then you sort them. Even if the endothelial cells are viable, like during the sort, after the sort, you put them on the plate, and the next day, all of them are dead. And I have no clue why, but it just doesn't work. So what we did is, or what they also did in the paper, is they cultured the lung homogenate, like the whole lung homogenate, completely undepleted, really just digest, put it on a flask, keep it there for a couple of days, and then go for cell sorting. And then it works. Somehow the cells need to be on a plate for one time, and then you can sort them and put it back, and then they're super happy and everything's fine. So I sorted just like single cells, and then I went for ABCAM negative, CD45 negative, ICAM2 positive cells. You might want to hear why I did not go for CD31. This is because CD31 is trypsin sensitive. And if you detach the cells from your flasks, you lose your CD31 signal. Of course, you can use other like enzymes, but I decided just to go for another marker. So I went for ICAM2, and this worked quite well. And then you can see that we ended up having a very pure population, actually. So we barely had any contamination from mesenchymal cells, epithelial cells, or immune cells. So we were really happy to see that. And then we checked again in flow, so checking for different markers for endothelial cells. So we checked for CD31. Here I used trypsin then to detach my cells. Checked for CD34 and VE Catherine, and they were positive for all of the markers, which we were really happy to see. And then when you put them on the plate, they do show the cobblestone morphology, which is typical for endothelial cells. And then we went for immunofluorescence to check for some more markers. So here again, we have CD31 and VE Catherine, but we also have endothelial nitric oxide synthase and VEGF receptor 2. So this was great to see that they do express all the markers. And then we were interested to see whether our endothelial cells actually expressed the markers for these two phenotypes I was talking about in the beginning, like the A caps and G caps. So the ones which are which express CAR4 or apelin receptor. And in fact, our cells do express both of them, which was great to see that even after some days on the plate, they still keep their microvascular phenotype. And as a last step before I went into the organoid cultures was to see whether my organoids are actually functional. So that's why I performed a tube formation assay. So I seeded my endothelial cells on Madrigal and just kept them on there for like 6 hours. And they formed tubes, which is very specific for endothelial cells. And I was super happy to see that they're also functional. And yeah, then we went into organoid cultures. And one thing we were concerned about is like the growth factors they need. And I mean, doing lung development, so once the lung starts to bud, hemangioblasts form blood lakes, which then start to sprout and form like the lung vasculature and the capillary plexus. And this VEGF is in the beginning released by the mesenchyme, and then over time it switches to the epithelium. And as we have like both compartments in our lung organoids, we wanted to see whether they actually release the VEGF already. And in fact, they do over time, even for 42 days, they do release the VEGF into the culture medium. So we were really happy to see that. So we thought, okay, likely we don't need to add additional VEGF because the concentration was already quite high. So what we did then was to sort IMCs and our BASC-enriched population from the adult mice and the endothelial cells from the pups, and then put them into inserts again to have an air-liquid interface and just have like triple cultures. Unfortunately, this didn't work that well. So in fact, the endothelial cells died over time. So this is day 21. And as you can see, there are no endothelial cells left, which was quite sad. But okay, we thought, okay, then maybe it's just not enough VEGF. So we added VEGF. And in fact, it helped a bit. So here in magenta, you can see the endothelial cells. But somehow they're mainly incorporated in the mesenchymal network and didn't really talk that much to the epithelial compartment. So yeah, we were a bit frustrated and we played around quite a lot. So we added different combination of growth factors like angiopoietin or VEGF and FGF2 whatsoever. Nothing worked. And then Monica had a great idea. So Monica was or is still a postdoc in Susanne Harold's lab, and she was the supervisor for my master thesis. And she had the idea to just microinject the endothelial cells into our organoid cultures, because during development, the endothelial cells or like the vasculature starts to form when the lung is already started to bud. So we thought it might be worth it to try to go for a later time point, but then as we have these Madrigal cultures, it's quite difficult to get them in there. So that's why she thought it might be worth a try to microinject them into those cultures. And that was the time where Ivonne actually became part of this project. So Ivonne is now my group leader here in Bonn, and she was doing a lot of microinjections in Gießen back then, and she was injecting mainly macrophages into the organoids. But of course, we don't want to have the endothelial cells in our organoids, but around. So what we did was to inject them around the organoids, so into the matrix or into the mesenchymal network, so that they weren't like swimming around, they were like in the matrix. And as you can kind of like sticking in here. So yeah, they're really just sticking onto the matrix or onto the mesenchymal network. And we have them quite close to the organoid, so they don't have to travel that far. And then we were super happy to see that after 21 days, the endothelial cells, in fact, really incorporated into our organoids, and they cover almost like 50% of the organoid surface, which was awesome to see. So we were really happy about that. And then we actually started to use that system, or like to do actual things with the system. But of course, in the beginning, we had to characterize it a bit more. So the first thing we were curious about is to check whether the endothelial cells still keep their microvascular phenotype. So we stained our organoids again for apelin receptor and for CAR4. And in fact, we were able to see that some of the endothelial cells are still positive for those two markers. And as you can see, the CAR4 ones, which are supposed to be the ACAPs, are kind of close to the alveolar compartment, and some of the cells which might be GCAPs are a bit further away. Of course, this needs to be investigated way more, but in the beginning, we were just like really happy to see that they actually keep their microvascular phenotype. And then in the next step, we were interested whether they're really talking with the epithelial compartment, because we already saw that they're like super close, but we didn't know yet whether they're really like communicating with each other. So we stained our organoids again for surfactant protein C for type 2 cells. And in fact, the endothelial cells like really aligned with the alveolar compartment, which was great to see. And they are really elongated and really just lying on top. And then, of course, we also had to check whether they interact with the type 1 cells. And it's kind of tricky to stain the type 1 cells in the organoids because they're super thin and really elongated. So what we did was we used protoplanin reporter mice. So the protoplanin was GFP positive. And then we stained our endothelial cells for the E-caterin. And as you can see, they also like really interact and lie on top of each other. Even though I know it's really hard to see here because like both cells are like freaking thin. That's why we asked our colleagues in the anatomy department for help, because they are really good in doing electron microscopy. So we sent them some samples, and what you can appreciate here is that, so here you have the alveolus. So this is a lot of surfactant that you have here. And then you have this very thin type 1 cell. And then you have the basement membrane. And on top, you have two endothelial cells. In red, you do see a connection between the two endothelial cells. And then the yellow stars mark the viable Palladi bodies, and showing that it's like really endothelial cells. And we were so happy when we got the results, showing that they're really like interacting with each other. So this was great to see. And then, as more or less kind of a last step, we needed to do kind of a proof of concept. So I mean, it was nice to see that they are actually like laying on top of each other, and it looked like they're communicating, but we didn't have like a proof that they're actually talking to each other. And what Ivonne did during her PhD was to infect the organoids with different kind of viruses. So this is what you can see here. So here we have an organoid, and Ivonne infected this organoid with a Cre virus. So all the cells that were infected became green. So this is just a normal organoid. It doesn't have like any endothelial cells. And as you can see, over time, the cells start to turn green. And this was actually an organoid from an MTMG mouse. So the cells were red, and once they were infected with the Cre virus, they turned green. And this is an alveolar region. And here you can see the cells become green, and then they start to die. And then, the alveolus is kind of gone in the end. So we decided to do something similar with our model. So we infected our organoids, and we decided to go for microinjection because we wanted to make sure that we disturb the endothelial compartment as less as possible, because what happens in vivo is that the virus comes from the inside of the lung, right? So the endothelial compartment doesn't get into contact with the virus in the beginning. So Ivonne injected the virus directly into the organoid, and she tried to really do it in the proximal region, and then the virus kind of diffuses to the distal region. So here you can see it. So here we did an NP staining for the nucleoprotein of the influenza virus, and very likely we injected the organoid somewhere here, and then the virus kind of traveled also to the distal region. So the infection was kind of spreaded. And to prove that the organoid or the epithelium actually reacts to the virus, we checked for pro-inflammatory cytokines. So we measured the IL-6 and the CCL2 release after 48 hours of infection. And in fact, both cytokines were released. And now the main question was, does the endothelium actually react to those pro-inflammatory cytokines? Because what happens in vivo is that the cytokines are released, and then the endothelial cells start to upregulate their adhesion molecules to allow the leukocytes to attach and then go into the lung. So we stained the organoids for AJRCMB. So in the upper row, you have the PBS control, in the lower row, you have the IV-infected organoids. And as you can see, in fact, they do upregulate the AJRCMB. So we were, I'm super happy to see that they really, that they really talk to each other, and that the endothelial cells start to upregulate their adhesion molecules. As a second proof of concept, we decided to go for LPS. So again, we injected LPS directly into the organoids, and it was shown that COX-derived prostaglandin E2 is released by the alveolar compartment, like the alveolar epithelium during injury. So we were interested to see whether this also happens like in our model. So we infected, or we stimulated our organoids with LPS, and then checked for the COX-2 expression. And in fact, the epithelium upregulates the COX-2, which then is supposed to have an effect on the endothelial barrier by the release of prostaglandin E2. And then we checked again for pro-inflammatory cytokines. So again, we went for IL-6 and CCL2. Here we checked after 24 hours, and indeed we did have an upregulation or a release of the cytokines. And then the last thing was, again, to check for the upregulation of the AJRCMB on the endothelial cells to see whether they actually respond to the pro-inflammatory cytokines. And in fact, they did. Compared to the PBS control, they upregulated the AJRCMB expression. And by that, we were able to show that they're really talking to each other and really communicating. And we were super happy to see that, because this is, as far as I know, the first model where you can really show communication between the different compartments. So to sum this up, we were able to integrate flow-sorted pulmonary endothelial cells into mature organoids, and this leads to the endothelialization of the organoids. And 2 weeks after the integration, the endothelial cells align with AC2s and AC1s. And they express markers which are known for microvascular phenotypes, even though we still have to investigate this a bit further, like if the cells behave like they do in vivo, like the different endothelial cells. And then we were able to show that LPS treatment and influenza virus-induced injury of the epithelium leads to a release of pro-inflammatory cytokines, which then leads to the upregulation of adhesion molecules on the endothelial cells, which demonstrates the epithelial-endothelial crosstalk. And the next step is to translate this into a human model, and this is what I'm currently doing in Bonn in Yvonne's lab. So I've been establishing an iPSC-derived, a human iPSC-derived long organoid model, and I'm currently trying to vascularize that. And just as a small sneak peek, so this is how it currently looks. So here we have a lung organoid shown in orange. So orange is the E-caterine, and in green we have the endothelial cells. And what is really exciting to see is that the endothelial cells form actual tubes, and they really align with the branches of the organoid. And yeah, we still have to characterize it a bit further. But yeah, we are super excited about it. And with that, I'm at the end, and I want to thank a lot of people. First of all, I want to thank you again, like the ATS team, for giving us the opportunity to share our data with you. And then I want to thank Ivonne and Susanne for giving me the opportunity to work on this project. In Yvonne's lab, a special thanks goes to Steffi, who helped me a lot with all the organoids and all the mouse work. And in Susanne's lab, a special thanks to Monika Heiner, who started this project with me and who gave me a lot of important input. And this work wouldn't have been possible without Yannis, who's running the imaging core facility in Gießen, and he helped us a lot with the optimization of the imaging process. So yeah. And then, of course, the people from the anatomy who helped us with the electron microscopy. And with that, I'm at the end, and I'm very happy to take any questions.
All right, thank you so much, Anna-Lena. That was beautiful work and really beautiful imaging. And we had actually had a guest, Bruce Stanton, on, and I was going to invite him now if he wanted to kind of kick off the discussion with the first few questions.
Sure. Thanks, everybody. Anna-Lena, that was really a terrific presentation. Congratulations to you on a really well done study, and also your team. It really exemplifies how doing these very difficult experiments takes a team approach. So congratulations. I look forward to hearing the work on the human iPS cells as organoid as well. Just want to make a couple quick comments. I think one of the important parts of this work is developing models on the lower airways. Most studies have been done looking at upper airways, the bronchial epithelial cells. That's one of the things that we work on in particular. But clearly, lower airways also, in just about every lung disease or infection model or system, do get infected. And there's probably significantly different responses of upper versus lower airway cells to these infectious agents. And I think this model is going to lead to a lot of really novel and interesting discoveries. The other advantage of using a mouse, of course, is that there are a lot of transgenics that were disease mice. And so you can do a lot of work that would be quite a bit more difficult. So we need better and more biologically relevant models, and this certainly is one. So congratulations. One other question, I'll just have one question. Have you guys tried to do inside out organoids? The reason being then, like you had mentioned in your talk, the bacteria, some of the viruses infect from the apical side, which to do those studies is quite labor intensive and difficult to try to inject into the sort of the intraluminal space. So that's my question. And again, congratulations on a really terrific study that I think is going to tell us a lot about lower airway function that we didn't know.
Thank you. In fact, we never tried it. We went for microinjections from the beginning on. We wanted to be able to just infect specific regions of the organoids, because if you add the virus to the medium, you infect kind of the whole organoid. And in fact, we realized that if you just add the virus to the medium, you can still infect the organoid. So you don't have to have it inside out. So it works quite well anyway. But yeah, we never tried it, to be honest. So yeah.
Thank you. I'll other people have a chance to ask questions.
Okay. There were two in the chat. If you want, I can read them, or if you can see also. The first was from Amali, and she asks, or they ask, how long before the culture dies after flu infection, given the lack of immune cells? And what are the kinetics of cytokine production and ICAM upregulation in the context of flu infection?
So how long it takes before they die completely depends on the virus dose. But most of the times they die quite fast. I mean, sometimes they are, if you give them like low dose, they are able to regenerate, especially if you add like additional mesenchymal cells, for example. But in general, they die after like 3 to 4 days if you give them a very high virus dose. Also, like the cytokine production, it increases, but like after 3 days, it goes down again. And to be honest, we didn't do kinetics on the ICAM upregulation. We just did it at this one time point, and it worked quite well. So we haven't investigated that.
Great. And then there was one more question so far. Elizabeth asks, do you also have experience with administration of bleomycin to the Vazquez organoids?
I that, unfortunately, no. Can't give you any information about that, sorry.
All right. I'm happy to ask a question unless anybody else has any for now. This is kind of a weird question, but I just was kind of curious if in sort of future iterations of this, you've kind of added the endothelium, but things like sort of flow and mechanotransduction are also very important. So is there a way you could see in the future adding that kind of next level of complexity to the system, since you've sort of gotten the cell type in place?
So with our system, this is a bit tricky because we are culturing our organoids in inserts. So applying flow is really difficult. So what we've been trying currently with the human ones is to have them like on an orbital shaker or just on a normal shaker in the incubator, and this helps a bit and it improves the endothelial network. So this is definitely a thing you can do, but apart from that, it's a bit tricky with having the organoids in an insert. And they really need the air-liquid interface, otherwise they don't differentiate that much. So yeah, a bit trickier than with other types of organoids, unfortunately.
Yeah, I can fully acknowledge that. I was just curious if you had already thought ahead at all. Okay, there's another question in the chat. So Clarice asks, have you looked at IL-33 upregulation after flu infection?
No, we haven't. And as far as I know, Ivonne didn't do it either during her PhD. So unfortunately, I can't give you any information on that.
We did try to look. I'm sorry. We did try to look at the IL-33 on their after giving the organoids dust, because we thought then there is a response towards the airway epithelium to then release IL-33. But surprisingly, we didn't saw it either. That was quite surprising because we do see that response from the airway epithelium, but not by IL-33. At least it's not mediated. But in influenza, we didn't do it.
And then the next question in the chat, is there a way to incorporate a microbiome, even an artificial one, for example, with a cocktail of antigens, to the lung organoid system?
I mean, introducing bacteria is always really tricky because they overgrow everything quite fast. Artificial one, I guess it should be possible. I mean, in the end, you can introduce more or less everything into the organoids via microinjection. But yeah, bacteria itself might be difficult because they just, you put them in the incubator and after 1 day you have tons of bacteria, but your organoid is kind of gone. So it's very difficult, unfortunately.
I had a question, Anna-Lena. That was some beautiful work, and I apologize. I'm not in this field. I work primarily with macrophage biology, but I was just wondering, I was sort of intrigued by the epithelial cell, endothelial cell crosstalk that you saw. And, do you have any hint, or do you have a sense of whether the presence of the endothelial cells also kind of impacts how the epithelial cells respond to an insult? Like, do you have any plans to do kind of parallel experiments, plus or minus the endothelial cells there, to see sort of the behavior of your model system in that sense and get a better idea of how that modifies it?
To be honest, we didn't do that yet. It would be definitely interesting to do that. To be honest, I would rather do this in my human system because it works better. But this is definitely something we would need to investigate before. Yeah, thank you.
I guess I can ask a quick question maybe. I study lymphatic endothelial cells, and I know in that context, monocytes and macrophages are sort of a major source of the VEGF, in this case VEGF-C, for lymphangiogenesis. Is that also potentially why kind of your system required a little additional VEGF?
As far as for the lung vasculature, it mainly requires VEGF-A, and this is mainly released by the mesenchyme and the epithelium. So I think the VEGF-C doesn't play such a crucial role here.
May I also then ask some questions? And really amazing work, really interesting data. And I have a few questions about endothelial cells, actually. Maybe the first one, it's about the picture where you show the staining for the CA4 and apelin receptor. Because they were kind of overlapped, I'm just was wondering, because you say one for the G-caps, another one for the A-caps, and I would assume that it shouldn't be like kind of staining for the similar for all the cells, but rather like this more CA4, this more apelin receptor, but in your culture it looked like more similar. Do you think they on the plastic become kind of the same type of cells, or what will happen?
We were also quite surprised to see that most of the cells seem to express both of the markers, but yeah, as you said, it might be due to the culture on plastic. This is very likely. I mean, to be honest, we were anyway quite surprised to see that they still keep the phenotype. So we were super happy to see that. But I guess, I mean, putting cells in culture and having them on gelatin or whatsoever always affects like the phenotype. So I would assume that this plays a role, yeah.
You didn't compare, for example, freshly isolated and after plastic. So like, it will be interesting how they behave. Then I have also another question. You mentioned also when you isolated endothelial cells, it was also from the pups. When for the epithelial cells, it's adult mice. Is it purposely done like that, or?
Yeah. So the thing is, isolating endothelial cells is in general a bit of a pain.
Yes.
And it's even more difficult to get enough endothelial cells from adult mice. So they seem to have more of a progenitor capacity when they're younger. So that's why I had to go for young mice. Yeah.
Okay. Yeah, kind of makes sense, I guess. And another question, sorry. I'm also was wondering about your ICAM1 data. So your endothelial cells were positive for ICAM1 after treatment. Nevertheless, I was wondering also, you have epithelial cells which are also supposed to be ICAM positive, and it was quite specific for endothelial cells, seems. Any explanation on that?
I am not so sure whether you were able to see that on the picture, but actually we do have some epithelial cells which also express the ICAM. It was tricky to distinguish like which cell is positive and which isn't. So we co-stained it for CD31 to make sure that we only quantify the signal on the endothelial cells.
Okay. interesting, because usually in my culture, because I also make a co-culture endothelial epithelial, I see that epithelial cells usually express much more ICAM. But this is human, maybe that's the difference. Could be human versus mice.
It might be, but we definitely had some epithelial signal there. Yeah.
Okay, but because that's something I would expect as well. Okay, the last question. I'm super sorry if someone else in the line, but it's just because I work in a similar thing, so I have tons of questions. It's very interesting trick when you put all the isolated cells on the plastic before you sort them, because we also struggle a lot with endothelial cell sorting. And I never thought about this. Nevertheless, I'm just wondering, when you put all the cells on the plastic, it's mix of everything, right? How do you make endothelial cells survive overnight? Or
So what I do is first of all I put them on gelatin cotton plates to support the endothelial growth, and then I also add some endothelial growth factor to the medium. I mean, of course, you propagate all the other cells as well, especially like any kind of fibroblasts. But I mean, I usually keep them for 2 to 3 days, so this is still fine. And if you just give them like the endothelial growth supplement, the endothelial cells grow.
And the fibroblasts don't overgrow, because usually those guys are crazy.
Yeah, I mean, they definitely grow like hell, but you still end up having enough endothelial cells, so it works quite well, actually.
Okay. Do you see that endothelial cells also proliferate during this time, or they just surviving?
Yeah, actually, it's really cute, because you can see that like over a few days you have these cobblestone morphology, so you can really identify the endothelial cells like within the lung homogenate. It's quite cool, actually.
Nice. Oh, that's really nice. Well, I'm really happy that you found this trick, because I really know that how complicated it is. Okay. Thank you very much. Very interesting.
Thanks. That was great. Good discussion. One more question in the chat. Isabel says, first, congratulations. This is a beautiful model. Did you have the opportunity to test the vasoactivity and contractibility within your organoid model?
No, unfortunately, we never tried that. This would actually be a great thing to do for sure. But yeah, no, we never did it.
All right. Anybody the floor is open for any further questions.
Erin, I have another question. You know, when we were offline, Dan had mentioned about fluid electrolyte transport in the organoids, especially in disease processes where that can really affect lung function. Have you had the chance to say, look at things like cyclic AMP stimulation, or PKA, or other aspects of fluid transport and the role of specific ion channels? I asked this. That's how I was trained, as an ion channel person, fluid transport person.
For sure, this would be super interesting to investigate, but unfortunately, we never did that.
I might add.
Can't do everything.
Sorry, what we do see in the organoids is like quite permissible to molecules, so you can really like diffuse molecules within the epithelial layer. So this could definitely be possible to do.
Yeah, as you know, people with rectal organoids use the rectal organoids taken from people with CF, cystic fibrosis, and they do swelling assays to determine whether certain people might be responsive to drugs, even though they don't have sort of the classic genotype. And so it's a nice screening platform looking at rectal organoids to look for the contribution of CFTR, and how basically therapeuting people with different mutations.
Sort of along those lines, do the organoids make tight enough junctions to block kind of diffusion between the cells, or are they more loose in terms of their architecture?
And in the organoid, you have they're super. They diffuse quite well. It's not like in the intestinal organoid. The junctions are super tight. Nothing passes through, and that's why I guess the need also to have these reverse organoids, and that we don't have that much with our system, because everything really goes through quite passively. Even when we macro inject things, of course, the organoids get inflated because of the liquid, and then the liquid just diffuse over time quite nicely.
Okay, thank you.
If there are any further questions, again, floor is open. Already had some great discussion too. Well, if that is all the questions for now, I'd like to just again thank you, Anna-Lena, for a beautiful presentation. It was really nice to get to hear from you, and thanks for coordinating the timing to be not as convenient for you, but a little more convenient for all of us here in the US. And really, really beautiful work. So thank you so much for sharing.
Thanks to you. Thank you for inviting us. It was a pleasure to share my data with you.
Of course, we'll look forward to more. All right, thank you guys so much. And this presentation was recorded and it will be uploaded. And so, yeah, for future reference, it should be available. Look out for that and share with others. And thank you so much.
Thank you.
Our journal club. And thank you, Bruce, for coming and discussing.
Thanks for inviting me. Appreciate it. Learned a lot.
Yeah. All right. Bye, everyone.